Welcome to our live webcast, Rapid Diagnostics in Clinical Microbiology as an Aid to Antibiotic Stewardship. Thank you for joining us. My name is Joy, and I'll be the operator for the presentation today. Before we get started, I would like to take a moment to acquaint you with a few features of this web event technology. At any time, you may adjust your audio using any computer volume settings that you may have. On the right-hand side of your screen, you'll see the text chat window. There's a large window which holds all of your sent messages and a smaller text box at the bottom where you will type in your questions. To send a question, click in the text box and type your text. When finished, click the send button. All questions that you submit are only seen by today's presenters and will be addressed at the end of the presentation. You will also be able to ask questions via telephone at the end of the presentation. At the conclusion of today's program, we ask that you complete a brief post-event survey. Please take a moment to complete this survey as it will help APUA plan future web events. We are joined today by our moderator, Dr. Barbara Lipinskis of APUA, and our speakers, Dr. Stuart Levy, founder and president of APUA, and Dr. Jerry Hall, our main presenter. At this time, I would like to turn the microphone over to our moderator, Dr. Lipinskis, for opening remarks.
Thank you, and welcome to our webinar. I am Dr. Barbara Lipinskis, and I am the Director for the Alliance for the Prudent Use of Antibiotics, APUA. I am delighted you could join us here today. I think you'll find the material interesting, useful, and with the attention currently being given to the topic of antibiotic resistance worldwide, it certainly couldn't be more timely. I do have to say that I'm proud that APUA has been raising the red flag of antibiotic resistance and urging antibiotic stewardship since our founding in 1981. So maybe the world is catching up with us. This webinar was made possible by an unrestricted educational grant for BacteriaScan, and we are grateful for their ongoing support. Now, our main speaker today is Dr. Geraldine Hall. Dr. Hall has held almost 37 years at the Cleveland Clinic's Clinical Microbiology Laboratory. She's taught at the Cleveland Clinic Medical School and has served as the director of the Medical Technology School at the clinic. She has a long list of publications to her credit, and she is more than eminently qualified to speak on the topic of rapid diagnostics. So we are very happy she agreed to do this webinar with us. Our topic today will be introduced by Dr. Stuart Levy. Dr. Levy is the founder and president of the Alliance for the Prudent Use of Antibiotics, and he is a recognized world leader in the area of antibiotic resistance and stewardship. He is also the author of the book, The Antibiotic Paradox: How the Misuse of Antibiotics Destroys Their Curative Powers. And now, Dr. Stuart Levy.
Today we are faced with many different kinds of bacteria and viruses that cause disease in our people. These viruses don't get up and tell us what they are. That's kind of hidden information. But we can identify the organism and parts of its being by using diagnostics, rapid diagnostic tests. In fact, there's energy being given to picking out the right treatment modality for respiratory, urinary tract, abdominal infections. It's a great help to the clinician, and it certainly has an effect on the emergence of resistance. Every patient who is missing the diagnosis and using the wrong antibiotic becomes a kind of factory of resistant organisms, which plague the physician and certainly plague the patient. We are looking for, and others too, rapid ways to identify the organism causing the infection, and then, and only then, to be able to arrive at a decision on the treatment.
Good day. Welcome to the APUA audio conference on rapid diagnostics in clinical microbiology as an aid to antibiotic stewardship. My name is Jerry Hall, and I'm going to be your presenter today. Hopefully you can sit back, listen to the presentation, come up with some great questions that hopefully I or someone else on the line can answer at the end of the presentation. I am calling you today from Forestville, New York, which I'm sure most of you have never heard of. It's a very small community, about 40 minutes south of Buffalo in western New York. It's my family's place of retirement. When my husband and I retired a number of years ago, this is where we moved, and I'll show you a picture of where we are in just a few minutes. But I want to welcome you to the audio conference, and hopefully, again, that you can sit back and enjoy what I'm about to present. Whenever anybody gives a talk on any topic at all, we need to start out with a list of objectives, so we're all sort of on the same page, knowing what we're going to be talking about for the next 40 minutes or so. And so on this first slide, what I've basically put down are 3 objectives. The first being, what I'd like to do today is describe the impact of existing and novel methods that are available for the rapid detection of bacteria. I probably will mention the word fungus on occasion as well, but most of the talk will definitely hone in on bacterial identification, rapid detection. Second objective would be to provide possibilities for the integration of rapid methods in all of our laboratories as opportunities to improve antimicrobial usage and to improve clinical and economic outcomes for our patients. Third, hopefully, we will explore the impact of existing and novel methods possible for the rapid determination of antibiotic susceptibility. So not only what the organism is that we've detected in a particular body site, but what antibiotics might we consider it to be susceptible to, or perhaps more importantly, what antibiotics might we consider that this organism might possibly be resistant to. So these are what objectives we sort of set out today. I want to make a comment at the beginning. I have really no conflict of interest to share with all of you. I do give some lectures, if you will, but in retirement those have diminished considerably. Indeed, I really don't have any conflicts of interest. APUA, APUA, the Association for the Prudent Use of Antibiotics, has graciously asked me to do this presentation today, and we're doing it in conjunction with a company called BacteriaScan. And I'll talk a little bit more about BacteriaScan and its product at the end of the presentation, because I think it's really one of these novel approaches, if you will, to not only detecting organisms, but also perhaps in the future determining antibiotic susceptibility / resistance. So those are the individual groups that I would like to thank at this point in time, but again make a comment that personally I have no conflict of interest that I need to share with you. So let's get started with this talk. What we wanted to do again is look at newer methods of identification, but before we do that, I thought it would be a good idea to look at what are the conventional methods for the identification of microbiological organisms. What do we do every day in our clinical labs to be able to give our clinicians information as to what's growing or not growing in the body fluids, specimens of their patients that they're taking care of in the hospital? So for identification, conventionally, we all are very familiar with manual biochemical testing. Now, I say that, however, many of you in the audience are newer medical technologists, perhaps, and you're not even familiar with manual because everything's become somewhat automated, even in clinical microbiology, but there is some manual biochemical testing that is certainly still done in clinical labs. We have continuous monitoring blood culture machines, lots of different types that are out there. Many of you are using these. Most of you are probably using these in your laboratories. Some of us in our laboratories screen our urine specimens to determine whether they're positive or negative quickly, so we can give our clinician answers quickly. Let's face it, what we are all about in clinical microbiology is doing the best testing we can do, most rapid testing that we can do, and sharing of that information with our clinicians as quickly as possible so they can take care of the patients from which these specimens came. In our laboratory, we have automated or semi-automated identification systems. And take your pleasure, whether it be Vitek or Microscan, Phoenix by Becton Dickinson, Thermo Fisher's VersaTrek, Biolog, or other systems that you may be using in your laboratory of an automated or semi-automated system. And if I don't mention one of the systems, it's ignorance on my part or just negligence on my part, but certainly fill that in for yourself. If you're using another system, that's working very well for you. But most laboratories are probably using one of these systems presently for conventional methods of identification. often the use of the manual, the blood culture machine, screening of urines, et cetera, this requires at least 24 hours before results are available about the identification of the organism. And if we're dealing with more fastidious organisms, it can even take longer than that. But we're kind of used to in our clinical microbiology labs not having an identification until 24 or maybe up to 48 hours. And I'd like to think by the end of this presentation or during this presentation, we all are fully aware of the fact that this probably isn't good enough. We need to get these results out faster than 24 hours, and it is possible to do that, at least for a good percentage of the organisms that we find in our clinical specimens. And maybe on top of that, we owe it to our clinicians and more importantly to our patients to be able to offer results even much, much less than 24 hours if indeed the specimen is totally negative, as in the case of a urine specimen. We know that a good percentage, and the majority of our urine specimens, are probably going to be negative. And wouldn't that be great to get those results out faster than 24 hours so that we can get patients on the appropriate antibiotics or perhaps on no antibiotics at all if they don't need them. So the conventional methods have worked. We sometimes become a little lackadaisical and say 24 hours is fine, but I hope that I will convince you by the end of this that we have other opportunities now to make those hours much, much less before we get that identification. On the next slide, pretty much the same thing. What have been the conventional methods for testing to determine antimicrobial susceptibility or resistance? Again, we have manual methods for doing this. We have the what some people would consider old-fashioned, but still a very good test, disk diffusion. We have e-tests. We have manual MIC trays that we can purchase, freeze-dried, frozen, whatever combination of the above you might be using in your laboratory. Or we have our automated or semi-automated methods for susceptibility testing. And again, you see the same list that was present on the previous slide with regard to susceptibility testing. And if you're using a Vitek or a Microscan, a BD Phoenix, a VersaTrack, often many of you are probably combining the identification and the susceptibility testing cards. Or maybe you're using them individually. Maybe you're waiting for the answer for the identification before you do the susceptibility test. However you're presently performing it in your laboratory now, this particular procedure, all of these, require isolated colonies. They're usually performed after the identification is completed, and this typically requires at least 48 hours from the time of specimen collection, and often more than that, especially if you're waiting for the identification before you perform the susceptibility. And certainly, I'm not recommending that one do susceptibility testing haphazardly before you even know what you're dealing with, because certainly the interpretation of susceptibility results does in many cases depend upon what the identification of the organism is. I'm sure all, or the majority of all of you who are listening in today, are using the CLSI guidelines for determining the susceptibility of the organism and interpreting the results that you get once the susceptibility is performed. Hopefully you're all following those guidelines or something very similar to that so that we are giving consistent results throughout all of our hospital institutions, et cetera. We do need some level of uniformity when we're doing this testing. So that's what the status of conventional clinical microbiology has been. I would say up until the last 4 to 5 years or so, maybe a little bit less than that, depending upon the size of your lab and how well you've adapted into some of the more rapid methods that have been developed. So before we get into some of those rapid methods, I wanted to use the opportunity now on a slide to talk about why rapid microbiological results are needed. And this is pretty much going to be not rocket science, but something that any one of you could have put together on a slide. So why do we care about getting results out more quickly than 24 hours or 48 hours? Well, certainly the overwhelming reason are our patients. We all want better outcomes for our patients. Yes, we all sit in our micro labs. Some of us even still sit in silos, hopefully not. But we do want our patients to get better, faster. We want lowered mortality rates on our patients. We don't want them dying. We want to give them appropriate, good targeted antibiotics as early as possible, not just throw broad spectrum antibiotics at them. not just throw antibiotics at them that we think should work or that clinicians think should work. We want to give our patients the opportunity to have less chances for more superinfections, so that deadly MRSA, that very unfortunate cases, those very unfortunate cases of C. difficile. We want to lessen the chances that our patients will have the opportunity to get these. So we definitely want better outcomes for our patients. I don't want to make this whole presentation about cost, but we are going to talk about costs, in particular, certainly cost to the patient. So we want for our patients the opportunity to spend less time in the hospital. We love them dearly, but we all know that the best place for a patient is home, recuperating, getting better after whatever ordeal they went through when they were in the hospital, but we don't want them to spend more time in the hospital because our results are taking longer to get to the clinicians and therefore to the patients. We want our patients to have fewer additional tests done. Now, when you look at a business plan, you say, wow, I want to do more and more tests. I want to charge more and more. Well, that's not really what we're in the business for. We want to do targeted tests. We want to do the best tests. We want to do them rapidly, and we want to provide our patients with information so that they don't have to have additional tests done because we've allowed the tests that we've done, the simplified tests, the first test, to give the correct results. Hopefully, and that's why I have a question mark here, if we do rapid microbiological results and we do present our patients with better outcomes and lower costs, that would include the use of fewer antibiotics, and with fewer antibiotics being used, fewer adverse reactions as a possibility. So these are all what I would call very good things and very needed things by which we need to embrace rapid microbiological results and, of course, do something with them, and we'll talk about that in a minute. Reduced length of stay in the hospital is an ultimate goal that we want for all of our patients, and as per the topic that we're talking about today, if we can reduce the development of antibiotic resistance by providing more rapid ID and susceptibility results for our patients, that would be a bonus, that would be our goal, that would be ultimately globally the outcome that we're all looking for. So we have some real big goals here in using these rapid microbiological results. And so I think the gist of this story that I'm telling today is we need to embrace these as much as we can. When are rapid microbiological results needed? Well, you know, in the big picture, all the time. If we can get results out fast on every test we do, why not? If they're accurate, if they provide better outcomes for our patients, why not? But certainly, the identification and susceptibility testing for blood cultures, for cerebrospinal fluid and other sterile sites, for cases of pneumonia, for those nasty wounds that the faster we treat them, the faster the patients can get out of the hospital successfully, for urine cultures, for sexually transmitted diseases, and I'm sure you can think of other situations where indeed rapid testing for identification and susceptibility would be important for us and for our patients. So there's all kinds of opportunities. In this particular talk, we're going to hone in mainly on blood cultures and urine cultures, but there are opportunities for rapid methods, for rapid results, for rapid communication in all of these situations and others. So is there data, are there outcomes data out there that support the idea that rapid does make a difference? So I want to present on the first two slides some what I would consider older data that, yes, indeed, rapid does make a difference. I'm sure many of you are familiar with the name Dr. Gary Dorn, a very good clinical microbiologist, still active, although retired. Back in 1994, Gary and his colleagues presented data looking at what at the time were maybe considered novel methods, these semi-automated ID/AST systems versus overnight. So we're looking at a Vitek, we're looking at a Phoenix, we're looking at a Microscan, and at the time, there was a limited number of these ID systems back in 1994 when you could actually get results before an overnight incubation or a manual identification and susceptibility test. And what Gary showed by looking at the semi-automated systems versus an overnight method was that the identification can come out in 9.6 hours versus the more conventional 19.6 hours. That's really a big difference. And again, this is back in 1994. We weren't looking at molecular. We weren't looking at MALDI. We were looking at semi-automated ID and AST systems. The susceptibility test came out in 11.3 hours versus almost 26 hours. And basically, what Gary showed in that study was that if you could get these results quickly, in his study, the length of stay of the patients remained the same, but the mortality was lower with the rapid results, 8.8% versus 15%. So perhaps the patients weren't getting out of the hospital any faster, but they were living longer. Mortality rates were lower. Good news. Fewer lab studies were ordered when he reported his data, and that's a good thing. The cost to the patient is going to be lower, and we're not going to have confounding factors coming on when we order tests that perhaps don't need to be ordered. And less time was needed for the implementation of appropriate antibiotics, as was shown in his study. So these are all very good things. Another wonderful microbiologist, pathologist, Dr. Joan Berenfinger, in 1999, presented an article in Journal of Clinical Microbiology where she and her group looked at an evening shift verification of VITEC results for the identification and susceptibility of organisms versus a more conventional, let's wait till the next morning, let's look at the results, and then let's report them out the next morning. What she and her group found in her study was that the length of stay for patients was 10% with this new rapid evening shift verification versus 12% with conventional methods of identification and susceptibility. Mortality was lower, about 8% versus almost 10% with the conventional methods. And she showed a cost savings, $4,900 per patient with the rapid evening shift verification versus over $6,600 with the conventional method. And she was able to show her administrators an estimated saving of $4 million a year by using these rapid methods. At that point in time, many laboratories were kind of fighting, if you will, with administrators about, should I be using these more costly identification methods? Why not just use the conventional? Does it really matter? And I think as I've shown you on these two particular studies that I've chosen to reveal, if you will, and you can go back and read them yourself if you choose, I think basically show that even with just changing an overnight read from a morning read, one can get some significant results both in patient care and in cost savings. Getting a little closer to now, Beekman et al. In 2003 published an article in Journal of Clinical Micro where they looked at 917 blood cultures. Most of the positive blood cultures were Staph aureus, E. coli, or coagulase-negative staph. The time of collection of the blood culture to a positive bottle that was then subsequently gram-stained was about 27.6 hours. They used continuous monitoring systems for the detection of these positive blood cultures and found that the time to notification significantly improved, both in decreased length of stay for the patient who had bloodstream infection and decreased length of stay after the diagnosis of bloodstream infection was made. So the quicker you get the results, the quicker you get information to the clinician, the more impact one has on the patient. And this was particularly with blood cultures. Dr. Berenfinger, again, in 2008, in an article in the American Journal of Clinical Pathology, went more to the basics. She went to the staining and the reporting of blood culture gram stains. And she was able to show that if the results of the gram stain of a positive blood culture were reported in less than 1 hour, a 10% mortality was shown in those patients or seen in those patients. As compared to waiting, not calling quickly, having a greater than 1 hour, reported in about 3.3 hours, almost a doubling of the mortality rate. These seem like common sense principles, use of ID, AST, semi-automated systems, getting the results out faster. But until we had information like this coming out in the data in journals like JCM, like American Journal of Clin Path, people were a little despondent, I think, and all of us were a little bit saying, oh, it's good enough to get it out at 24 hours. It's good enough to get it 48 hours. It doesn't make a difference. Well, yes, it does. The older data shows that, and I'll show you newer data that also supports the fact that the more rapid you can get the results out and communicate it to clinicians and hence on to patients, the better it is for all of us. So what are the newer methods for identification? Do we have opportunities in our laboratory to speed up the time it takes to get an identification and a susceptibility? Sure. So what's come on the market real recently is a wonderful instrument. I know you're all familiar with it. I don't need to go over the principle of it, but MALDI-TOF. So the use of an instrument, the use of a principle which allows us to get identification, usually of colonies, but as you're all aware, also being able to use MALDI-TOF directly in blood culture bottles, for example, that are positive, to be able to get answers in minutes to hours as compared to overnight, as compared to even an identification semi-automated system, which might take 6 to 8 hours or more. So MALDI-TOF has certainly revolutionized the identification of organisms in the microbiology lab, not only bacteria, but fungi as well, mycobacteria as well, and other organisms. And I'm sure there's just tons more data that will come out over the next few years showing where we can implement MALDI and how we can make it useful to us as a rapid method of identification. Sequencing. I don't know how many of you who are listening online today have sequencing methods in your laboratory, but certainly the use of sequencing methods allows us to get rapid identification, more so than we were ever able to do in the past, and very accurate identification of organisms. In addition to MALDI and sequencing in our laboratories, there are methods for direct specimen testing, whether we use probes or we use nucleic acid amplification testing on our blood culture bottles, on specimens directly. And as I'll mention in a few minutes, there are even some methods that are being employed in a research format right now for direct blood testing without the need even for continuous monitoring blood culture systems. That's really going to revolutionize the world when it becomes more available to we in our clinical laboratories. And I'm sure you can think of other newer methods for identification, and we will mention them as the talk goes on. But these are some of the ways in which, in our laboratories right now today, we can implement and achieve rapid methods. And some of you will say they're too expensive. I can't change the way I'm doing things. Our administrators would not allow us to do it. But we have ammunition. We have articles, and I'm going to show you some more outcome studies that have been done with some of these methods to indeed be able to show our administrators, our clinicians, our patients, that with rapid methods, we can move forward to get better patient outcomes and reduce antibiotic resistance development. Do we have any newer methods for antimicrobial susceptibility testing? Absolutely. We can now use methods in our laboratory to detect resistance markers. I've just mentioned a few here, and I don't mean to slight other companies because there are so many. Verigene, Cepheid Expert, MRSA using genome, other methods of detecting resistant markers, whether that detection be from a colony on a plate or a positive blood culture bottle or even directly in clinical specimens. Now, there are some questions with the use of detection of resistance markers, whether it be with a sequencing or some other method, as to whether the genotypic or molecular detection will be equivalent to or be the same as our more traditional, conventional, slower phenotypic methods. And that's still a question which needs to be answered. The fact that an organism has these markers, the fact that it has potential to develop resistance, doesn't necessarily mean it will. Doesn't mean it will express it. Do we have to continue to use our so-called old-fashioned phenotypic methods? Or are there opportunities for newer, quicker phenotypic methods to be employed? And we'll talk about that in a little bit when we talk about bacterioscan and some other methods that can be utilized in the laboratory potentially in the future. But these newer methods for the detection of resistant markers are a huge start, and they are a huge addition to our rapid clinical microbiology methods to give our clinicians a better handle on whether the organism that has been isolated from their patient's specimen is of the wild, susceptible type, or does it have the potential to be resistant to drugs that we would usually use to treat it. And this is just wonderful news for all of us in the clinical microbiology labs. I personally think the need is for identification and susceptibility to be keyed together, and we need to work on rapid methods and continue to work on rapid methods for both of them. But let's face it, what we really need to know for our patient is this organism going to respond to the typical drugs we might use, or do we need to use something different? So let's get into the gist of this. How can one employ rapid diagnostics? And we're going to particularly look first at bloodstream infections. So the easiest, I would say, way to look at this is if we know it's going to take up to 24 hours before we identify an organism in a blood culture from a routine continuous monitoring system, and then more time, obviously, for susceptibility. Can we do something more rapid? And the answer is yes. So direct testing from a positive blood culture bottle, which has been keyed as positive on your instrument, can be done today in your laboratories. By use of fish probes, for example, fish probes have been on the market for quite a long period of time, actually. There's been a number of outcome studies. I didn't include them in here. Perhaps I should. If any of you are interested, I can certainly send you references to those articles on fish probes. But you can Google, you can go to PubMed, and you can look up articles on your own as well, and show that there have been many outcome studies showing that with the use of fish probes for the identification rapidly of MRSA, of enterococci, of coagulase negative staph versus Staph aureus, of some very significant blood culture pathogens of the gram-negative nature, such as Klebsiella and E. coli, Pseudomonas aeruginosa, and certainly fish probes for yeast, have been shown when they're used appropriately, when they're used rapidly, and when the information is rapidly communicated to our clinicians, their impact on patient care can be huge, both on length of stay, reduced mortality, and cost to our patients in a reduced format. MALDI-TOF, as we mentioned, can be done directly from positive blood cultures. And certainly getting an answer of identification by MALDI-TOF in minutes to hours, as compared many times to overnight results, would be wonderful for our patients. I already showed you in early data that with these semi-automated systems of identification, one can get better results more quickly to our patients to assist in their decreased length of stay, decreased mortality, etc. Rapid MRSA testing. We all know that MRSA is a big-time pathogen. Being able to let the clinician know that their patient has MRSA and not MSSA is going to save their lives, is going to get them better antibiotics, is going to reduce the tests that have to be done. So if we can do this rapid MRSA testing, why not? Verigene and other film arrays that are out there now and available in your laboratories, in my laboratory, can be used to identify specific organisms and resistance markers in some of those organisms as well. So we do have a number of methods that can be used today in our laboratory for rapid diagnostic for the identification of bloodstream isolates. Now, something perhaps even more exciting is what's being done on a research basis for direct testing from whole blood. So what this means is we don't necessarily have to wait for the blood culture bottle to pop positive. We don't have to inoculate blood culture bottles. We can actually do testing directly in whole blood. Many of us who are as old as I am know that we've thought about this in the past. Wouldn't it be great to just stick a patient, get blood, test it, and say right then and there, within an hour or less, this is what your patient has in their blood culture? There have been a lot of negatives, a lot of pessimism, a lot of thought that we'd be picking up too many coagulase negative staphs, whatever. But there are a number of products, and I've listed two of them for you here, that are being used particularly outside of the United States, mainly on IVD or research basis, some of them homebrew. One is the Light Cycler Septic Fast by Roche, a broad-range PCR in an IVD format. I've given you a reference of one article that talks about how this can be used and significantly reduce the time to identification of organisms directly in whole blood. Cesti test is a home brew out of Germany. I've given you an article if you'd like to read about that and to show what it can do, both, again, being able to identify organisms in whole blood. Are they 100% accurate, 100% sensitive, 100% specific? Probably not, but then you have to agree with most of the tests that we do in the laboratory. There are very, very few that are 100%, if any, that are 100% sensitive and specific. But we want to be able to get the fastest results we can in the most accurate format as best we can. There's some research being done in the United States on a product called T2 Candida that identifies the majority of significant candidal isolates that we find in blood cultures in a very rapid approach, directly testing whole blood. So these are things to look out for in the future, but we don't have to wait for the future. We can do direct testing from our continuous monitoring blood culture systems right now with those tests that are mentioned on the left and others that are about to come. Many of you may have been at the recent ASM meeting. I unfortunately did not get the opportunity to attend this year, but I know from talking to some of my colleagues, there are a number of products, a number of companies which are coming out with direct testing from positive blood cultures and direct testing in other clinical specimens. And so all I can say is keep abreast of what's going on. If you haven't been able to get to the ASM meeting, you can certainly read information and go to the ASM portal and get information about what was said and what products are coming out. Talk to your colleagues. Listen to webinars such as this one to gain more and more information. And use PubMed, use Google. Use those systems that you think not everything on Google is right. I agree with that. But use what you can, the CDC sites, of the PubMed sites, to be able to read the literature and find out what products are coming out and what more and more rapid systems are available for you to use in each of your clinical labs. Now, I want to step away from the topic of rapid diagnostics just for a second and show how they apply to the concept of antibiotic stewardship. I know that this is not a new term to all of you. I don't think anybody needs the definitions I'm going to give you, but I'm going to do that just so that we're all on the same page. So what is antibiotic stewardship? It is a multifaceted approach to optimize the use of antibiotics while at the same time minimizing the development of resistance and other adverse effects that come with resistance and not using the appropriate antibiotics. If you look up the definition of stewardship in the dictionary, you find that what it is the acceptance for responsibility for long-term management of something of enormous value. And what is of more enormous value than doing better patient care, doing better laboratory work, and stopping resistance development? And that's what we're all about. I mean, again, get out of our silos, know that we are taking care of patients, I know you all know that, and accept some responsibility for doing a better job. Good stewardship practices is an active and dynamic process of continuous improvement on antibiotic usage. It's an ethic. It's a way of life. It's a way we do our business, with many steps of different sizes by all who are involved in antibiotic use. And that's all of us. A prior term that was used, so it's not, antibiotic stewardship showed up maybe about 10 years ago, and some people jumped on it, others did not. People assumed they were doing antibiotic stewardship, even if they didn't really use the term. And a prior term that was used was prudent antibiotic use. And I think that's still a great term, and I love that APUA actually uses that term, if you will, of the prudent use of antibiotics. But antibiotic stewardship, another name for the same thing. We all want to do a better job. We all want to reduce the use of antibiotics when we don't have to use them. We want to use the right antibiotics when we should be using them. And that's what we're all about. So before we talk a little bit more about that and more about rapid methods, if you will, does it really matter? We talked before, does it matter if you get rapid results? Well, I've shown you in at least 4 articles, yeah, it does matter. I'm going to show you a few more in just a little bit. But do resistant organisms cost more than susceptible organisms? Cost to the patient, whether it be in their length of stay or actual cost, if you will. Does it cost more if you have a resistant organism? So there was a study by Nordell in 2002 that was published in the Clinical Infectious Diseases. I've given you the reference. And basically what the study looked at was methicillin-resistant Staph aureus, vancomycin-resistant enterococci, Klebsiella that were resistant to the carbapenems, Pseudomonas aeruginosa specifically resistant to fluoroquinolones, and Acinetobacter baumannii, which was ampicillin sulbactam resistant. And so the authors looked at these specific organisms and said, does it cost more to have a resistant organism? And what they found that if the patient had one of these resistant organisms versus MSSA, VSC, et cetera, there was a 7-day increase in the length of stay of those patients in the hospital. Even when they got rid of all the confounding factors that would make patients stay in the hospital longer, patients with susceptible organisms were in the hospital 1 day less than those with resistant organisms. So it looked like, yeah, we can reduce the length of stay, if a patient has a resistant organism, they're going to be in the hospital longer. And since the title of this slide is cost, there were $70,000 in increased charges associated with patients who had resistant staph versus susceptible staph, resistant enterococcus versus susceptible enterococcus. And again, when confounding variables were removed, the difference was about an $8,000 in increased charges. So yeah, it does cost more. We can show that. And there are other studies that have actually shown this as well. In addition, maybe more importantly, the death rate was higher for hospital-acquired infections caused by vancomycin-resistant enterococcus or imipenem or other carbapenem-resistant Klebsiella as compared to community-acquired, which of course tended to be more susceptible. In addition, the study showed that pneumonia and urinary tract infection were also the cause of higher death rates. So keep that in mind because we're going to talk about urines in a bit. And lastly, the community-acquired vancomycin-resistant enterococci patients resulted in a $69,000 higher charge than if that patient had vancomycin-susceptible enterococci. So yeah, it does appear that resistant organisms cost more than susceptible organism infections. I think that makes sense to all of us. You're in the hospital longer. It's going to take changes in drugs. If we don't do rapid results, we're not going to get the results out that say that this organism is resistant. So one of two things is going to happen. Patient's going to be put on broad-spectrum antibiotics, which may cause even more resistance development, or they're going to be put on inappropriate antibiotics. So enough said, I think, about that. But indeed, yeah, there is a significant difference between having a resistant organism or a susceptible organism. Getting back to the idea of this antibiotic stewardship and how we all can help in the laboratory, the Healthcare Infection Control Practices Advisors Committee, in partnership with the U.S. Department of Health and Human Services, has listed antimicrobial stewardship as one of the top 5 messages for healthcare workers in 2009. So the word is, whatever you can do towards antibiotic stewardship, reducing use of bad drugs, reducing overuse of drugs, et cetera, this is what we need to be doing. And this was back in 2009. We're now in 2015. What have we done in our laboratories to increase antibiotic stewardship? Think about that. MRSA infections were listed as one of the 6 categories of healthcare-associated infection for the Healthcare Infection Control Practice Advisory's 5-year national prevention target. The cost of Staph aureus bacteremia, $20,000 to $70,000 an episode. If we can reduce this, we can reduce costs and get better patient outcomes. So what are some good stewardship practices in clinical microbiology that we all should be aware of? Well, I found this article by Prescott et al. In a journal called Veterinary Microbiology. Go figure. But when I Googled and when I went to PubMed, I found this article, and actually in the title, it had the tsunami of antibiotic resistance in its title. So it caught my eye. And I read the article. And when I read the article, I came up with a list of these good stewardship practices that were listed in the article, and I thought I'd share them with you. And this is something we all can do in our clinical labs. Antimicrobial use guidelines and categorization. We need to develop good antibiograms that we share with our clinicians. We need in our laboratories to have usage guidelines when we're reporting out our antibiotics. We need to categorize antibiotics based on the lowest possible denominator, so to speak. If Keflex works, why not? If we need to go to a higher class drug, then we should. But we don't need to report out every single drug that's tested in our laboratory necessarily if the organism is susceptible. Now, that's going to change as we use some of our more rapid methods, as we'll talk about for susceptibility testing. But keep in mind, we still need to provide antibiograms. We still need to be part of the antimicrobial use guidelines development within our hospitals, we being clinical microbiologists. In hospitals, voluntary restriction policies are absolute and necessary. Not every drug that comes out on the market, although there are certainly fewer and fewer of them every year, but not every drug should be available to every patient, to every clinician, and there should be some restriction policies. Formularies need to be developed with appropriate drugs listed there for patients to be given, for clinicians to be delivering, and so that there's some semblance of stabilization, some semblance of organization and cooperation among all clinicians with what can be used and what can't be used. Test utilization is a big buzzword in clinical microbiology and throughout hospital systems. Just because you can order a test doesn't mean it should be ordered. We found at our hospital, with a very large committee of individuals who were working on test utilization, that sometimes patients were ordering C. difficile 2 and 3 times a day. Sometimes physicians were ordering tests for genetic testing on patients, DNA testing, which doesn't change in a patient's lifetime, more than one a week, more than one a patient's lifetime. We need to be aware of when tests should be ordered and when they shouldn't be ordered, and when stop orders should be put onto clinicians so that when they order a test that's just been ordered, maybe by somebody else, they know it and they're told, no, you can't order this again today. It's already been ordered. You only need one a day. If your patient's been in the hospital longer than 3 days, don't order a parasite on a stool specimen unless you have some really good reason for doing that. We need to have control of what tests are being ordered by our clinicians, not as a punishment, but as an education. Education is, of course, the biggest buzzword that we all should be living by, and we need to provide information to our clinicians about what we think are the best tests to order, what we think are the best drugs to be used based on the tests that we're doing in our laboratory, and they indeed need to educate us about the clinical conditions of the patients and supply us with more information about what indeed they are suspecting their patients may have as a disease situation, if you will. So education is key to all of good stewardship practices. The regulation of access to antibiotics, along with the development of formularies and voluntary restriction policies, is absolute. All of us in the clinical world have to deal with conflict of interest issues. I told you in the beginning of this talk I felt I had no conflict of interest with talking about all of this. I don't work for any of these companies. I'm not consulting for these companies, et cetera, et cetera. But I think all of us have to be aware of the fact that conflict of interest does come into our lives, and we need to, as I said, be aware of it, but also deal with it and have some real good regulations within our hospitals about it. And first and foremost, we need to implement, all of us, good infection control practices, whether that be hand washing or whether that be taking care of our patients by isolation, et cetera, depending upon the infections that we may have helped to find in them, if you will. So these are good stewardship practices that need to be accomplished by all of us, both in clinical microbiology and outside of clinical microbiology. But I really do offer you that article by Prescott et al. Take a look at it. I know it's in veterinary micro, and one might say, what does that have to do with us? But it has a lot to do with us. So who should be involved in antibiotic stewardship? So these are coming up on your screen. Infectious disease clinicians, of course. We clinical microbiologists, ID pharmacists, infection control practitioners, the patients themselves, and our industrial colleagues, whether that be companies that provide medical devices or instruments or our pharmaceutical industry. All of these individuals, and you can probably name some others, need to be involved in antibiotic stewardship. And we would do well to know these individuals in our hospital, to know our ID clinicians, of course, to get to know your ID pharmacists. They can be a big help to you, especially when we talk about communicating results. And I would look at all of our industrial colleagues, as friends. Not that they're going to provide conflict of interest for us, but they are individuals who can provide education back to us and we to them. So, how do we provide rapid results? That was the gist of this talk. What are we going to do? Well, our goal in the clinical microbiology lab is first and foremost to communicate the best methods for specimen collection. We are about getting good specimens in our laboratory and providing good results. A sort of go by the mantra of garbage in, garbage out. So we want to make sure that we tell our clinicians and our patients when necessary what's the best way to give us the specimen that's going to give the best answers. We want to use methods that will provide the quickest and most accurate results for the good old gram stain. That's not going away. When we talk about rapid results, what can be more rapid than reading a gram stain, than seeing whether there are white cells on there, seeing whether there are specific predominating organisms on there? We need to have quick and accurate results for ID, for susceptibility. Don't forget about serology and antigen testing, and certainly direct specimen testing when we can do it, when we can provide it, whether it be for an STD or a direct blood specimen testing. If we can do that in our laboratory, we need to take the opportunity to get those quick and accurate results. And perhaps most importantly, we need to report and communicate those results as quickly as they are completed. Going back to the Barenfinger study looking at gram stains, when she turned around her blood culture results in less than an hour, she significantly impacted patient outcome, or her lab significantly impacted patient outcome, rather than saying, oh, I can wait an hour or two, I can batch them, I can call them later. We want to communicate those results by phone, by email, electronically on our lab information systems, or all of the above. We can quibble about how often do we have to call the same positive result from a blood culture. We can quibble about how often do we have to call that STD result, if you will, or that C. diff result. But remember the patient. Remember the data that shows if you get the results out more quickly, patients get on right antibiotics. They get out of the hospital faster. They don't die as quickly or as much. So we really do want to do all that we can, as efficiently as we can, to get our results out. Remember, the results that we have in our hands in the laboratory are no good to our patients until we get them to our patients, to our clinicians. So I wanted to present a study that was done looking at rapid diagnostics in bloodstream infections in 2014 and how that affects antibiotic stewardship. Kofari et al. Presented in Clinics in Infectious Disease a study looking at, how fast do we need to go, how fast do we need to get results. And what they showed with particular respect to bloodstream infections was that there was a mean decrease in survival of their patients of 7.6% per hour after the onset of hypertension until effective antibiotics are on board. So the longer you wait to get the right antibiotic on board, you're increasing the chance for that patient to die per hour by 7.6%. I don't mean you specifically, but I mean the system. The use of inappropriate antibiotics was shown in the study within the first 6 hours after recognition of septic shock was associated with a 5-fold increase of mortality rate. So if we're not able to get the right antibiotics into the patient, or the clinician's not able to get the right antibiotics in, you can be up to a 5-fold increase in mortality if not done within the first 6 hours, getting them the appropriate antibiotics. And we know now that we can help. If we can get that susceptibility test out faster than what our conventional methods can do, we can assist in decreasing this mortality time. In addition, what Kothari et al. Showed was that the answer then, by many clinicians, knowing this data, would be, let's just throw a lot of antibiotics. Let's give our patients imipenem and ciprofloxacin and cephalosporin. Let's just throw the whole wall at them. But what's that going to do? That's going to increase antibiotic resistance. That's going to reduce antibiotic stewardship. That's going to really throw a ringer into everything if we just give more antibiotics. That's not the answer. And we all know that. That's what's been done because in many cases we have not been able to provide rapid answers. So on the next slide, what Kothari did in that same article, and I have it in a chart here, and we don't have to look at every point of this, and you can look at this at your leisure, but what they showed in their article was that if direct testing from a blood culture bottle, whether it be by fish or MALDI-TOF, GeneXpert, or Veregene was done. And then one looked at mortality, changes in length of stay, cost savings per patient, and looked at that in regard to whether there was an antibiotic stewardship intervention, whether somebody was called early, whether somebody was, a clinician was physically called by an ID pharmacist and told, you need to change the drug, that's not the right drug that your patient is on based on what the laboratory is showing us. If you go down this list of the studies that Kothari et al. Looked at, when fish probes were used in the first 4 studies, there was no mortality benefit that was recognized, or at least it was not determined in a couple of the studies. But when it was determined, you can see in the fourth one down, that there was actually a 16.8% benefit versus 7.9% mortality benefit, if you will. So a reduced mortality when the fish probes were indeed used. There was a change in the length of stay, and there was a cost savings, although, as you can see, the difference did not appear to be statistically significant in those particular studies. And on the very right-hand column, you can see that whether or not an antimicrobial stewardship intervention was done. So in the fish studies in particular, when the antimicrobial stewardship intervention was done, as in the first study, 2 days less length of stay and a cost savings of $4,000. When there was no intervention, there was a slight change in the length of stay, or a similar change in the length of stay. But note what happened. There was actually an increase in the length of stay without the antibiotic stewardship intervention. In the third study, cost savings to the patient with this stewardship intervention, and I already mentioned the fourth study with the fish probes, when indeed there was no intervention that was really done there for antimicrobial stewardship. So bottom line on this one is, if you're going to do the rapid testing, you've got to communicate the results. You've got to really let the clinicians know that, this is what my data is showing. It is rapid, here it is, and you need to change your drug. GeneXpert MRSA versus Staph aureus, an antimicrobial intervention done. In many of these studies, the ID pharmacist helped out. And I'd like to put a plug in for that, because I think if you can work with your ID pharmacist to help you make some of these phone calls, make some of these interventions, they're going to have a lot more clout with regard to antibiotic changes. And so what you can see is a 6.2-day less length of stay in the hospital, $21,000 savings to the patient. With MALDI-TOF, when an intervention was done, you can save length of stay in the hospital, you can save some money, and you can benefit the patient with lowered mortality rates. With Veregene, this was basically only with yeast, but with intervention, there was a considerable cost savings to the patients and almost 22 days less length of stay in the hospital. So what Kothari et al. Showed is basically, if I do implement these rapid testing and I do intervene along the lines of antibiotic stewardship and letting clinicians know they should be using other drugs, et cetera, I can make a significant difference. So you can go back and review that article if you'd like to as well. What about specifically methicillin-resistant Staph aureus bacteremia in rapid testing? Well, there was a study done at Ohio State University looking at the expert MRSA assay compared to conventional methods employed in bacteremia with methicillin-resistant Staph aureus. And they employed a partnership of the microlab, epidemiologists, the pharmacy, infection control, and infectious diseases. And what did they show? The length of stay of the patient was over 6 days less with rapid methods, cost the patient $22,000 less, and the results were 100% sensitive and specific as compared to the conventional methods they were used. So they got good results, they got accurate results. When they employed a partnership of all of these individuals, they were able to extract a very good patient outcome just with one organism testing. And the end result in their article was that the ID pharmacist was essential. And again, I implore all of you to know who your ID pharmacist is, get them on board with antibiotic stewardship, which of course they're a part of, and get them on board with helping you with reporting the results that you're getting out of your laboratory more rapidly. So we talked a little bit about identification and how can we speed that up. We talked probably a very little bit about susceptibility testing and how we can speed that up. So I wanted to talk some more about that. So there are some newer technologies and methods for rapid antibiotic susceptibility testing. We can use, or the availability is out there to use some of these newer methods. So these are still in the stages of research. These are still in the stages of being reported in the literature, in specific laboratories or research institutions, but they're coming. You need to be aware of them, and you need to be looking into them. So there are some imaging techniques. There's a system called Baxtel, called Accelerate Identification and Susceptibility Testing. Right now, presently, it's being manufactured or worked on in Tucson, Arizona. It's in an RUO format, and it's for use in positive blood cultures. So you can actually get results more quickly than our traditional identification and susceptibility using imaging. And look out for that. We've all heard of FACS before when we talk to our hematology colleagues. This is fluorescence-activated cell sorting, which alone or in combination with microfluidics, and you'll hear more and more about microfluidics if you read the literature about rapid testing for susceptibility, to distinguish cellular size and shape. And fluorescent stains can indeed distinguish dead from living cells. So by using a FACS instrument, by using organisms that are subjected to antibiotics of different types, these stains can distinguish dead from living cells and help us to show whether or not an organism is going to be susceptible or not to a particular drug, much more quickly than overnight incubation or our conventional methods can do. Microbial cell weighing by vibrating cantilevers. This is a really catchy title here, but this is a possibility for performing rapid antimicrobial susceptibility testing. Microcalorimetry, measuring different heat production by organisms in the presence or absence of in the presence or absence, if you will, of antibiotics, to be able to show whether or not we can indeed determine resistance to various antimicrobials. In addition, on the next slide, rotating magnets that will attract organisms based on whether or not they are susceptible or resistant to a variety of antibiotics. RNA sequencing to pick up markers for susceptibility testing. Testing in microdroplets, so you're using minute amounts of organisms, minute amounts of antimicrobials, and being able to get results much faster. A number of years ago, there was some literature about adapting bacteriophage susceptibility testing to more quickly predict whether Mycobacterium tuberculosis will be susceptible to drugs like INH, rifampin, etc. There was a flurry of activity about the use of bacteriophage susceptibility in TB organisms, and then the literature sort of died off. But this is again being developed for non-tuberculous bacteria, for bacteria that we're most associated with in bloodstream infections, etc. And so look forward to that in the future, possibilities for more rapid susceptibility testing. And there will just be a gamut of others. Now, I refer you to the article from which I got this list, Van Belkom et al., in the Annals of Lab Medicine in 2013. It's a wonderful article talking about newer technologies for both ID and rapid susceptibility testing. And some of the downsides, the fact that you use a rotating magnet to show that the organism might have reduced susceptibility to a drug, is that going to equal what the phenotypic results would result in, et cetera? But I think it's worthwhile, all of us reading articles such as this, being aware of what the possibilities are and seeing where they may fit into our own respective laboratories. So yes, there are some newer technologies that we can look forward to with regard to rapid susceptibility testing along molecular and other lines. Will rapid diagnostics alone reduce antibiotic resistance? Is all of this just a lot of gimmickry, if you will? Do we need to have these rapid methods, if you will? And are they going to work on their own? So what I'd like to present to you here is the fact that, no, we need rapid diagnostics, but we also need a better understanding by all of us of the emergence and speed of resistance development. We can, again, just sit in our laboratories and do our testing and not particularly understand what's going on. We need significant development of new antibiotics. I don't think any of you in the audience are unaware of the fact that we have not seen a lot of new antibiotics coming out over the last 10 years. When I first started in clinical microbiology almost 37 years ago, we were seeing many antibiotics coming out. We had all these cephalosporins, all these penicillins, fluoroquinolones started coming out. It was like, wow, how can we keep up with all of these? How do we know which ones to use? Well, there's been a tremendous slowdown, if you haven't noticed, in the development of new antibiotics, and we need that to change. We need to share global surveillance data. You need to develop your antibiograms. You need to compare them to what's going on in your region, throughout the nation, and throughout the world. You need to know what's coming. You need to know if there's some resistance that's developed in a country outside of our own. What's the chance that it's going to hit us? What's the chance that we're going to see it? Imitanum, or rather carbapenem-resistant Klebsiella, is a great example of that. There's just been an article recently about some Shigella that are resistant to drugs that they've always been susceptible to. We need to be aware of all that. We need to know what's possibly coming. We definitely do need to increase the development of the technical needs for a technologist. All of us need to be brought into the picture of large-scale data management, bioinformatics, how best to communicate this information. We can't sit in our silos in the laboratory anymore. We need to share that information. We need to be aware of what's going on with regard to antibiotic resistance. So getting better methods of detection is wonderful. Grasp it. Keep abreast of what's out there. Bring it into your laboratories. But also educate, educate, educate yourselves. We've talked mainly about bloodstream infections and about how rapid can certainly help with the diagnosis of those infections. What about other body sites in rapid testing? And in particular, I'm going to spend a little bit of time talking about urine. So first of all, what's the reason for a urine screen? Why do we need one of these in our laboratory? Well, it can provide rapid identification that the microbe count exceeds the threshold, whether the threshold be 10 to the 4th or 10 to the 5th colony-forming units per ml. You can reduce the number of urines that need to be plated in red. This is going to save personnel time. It's going to save money. You're going to be able to inform the clinician about whether that urine is positive or negative in a more rapid period of time, and therefore the individual physician is going to be able to use appropriate antibiotics sooner. Or, on the other hand, if the urine screen is negative, reduce the use of antibiotics totally and not use unnecessary antibiotics. Interestingly enough, when you look at bloodstream infections in the laboratory, the most common source from which these bloodstream infections occur is a urinary tract infection. So just think about this in the hospitalized patient. If we can get the answers out earlier about whether or not a patient is positive, i.e. has a urinary tract infection, or whether they're negative, in a much more quick approach, using some of these more rapid diagnostic systems, if you will, we're really going to then perhaps help to reduce the amount of subsequent bloodstream infections that occur in these patients. And wouldn't that be great if that happened? So what are available urine screening methods? Some of them have limited success. Some of them are accepted. Some of them are not. Certainly the gram stain has been used in order to screen urines. Dipsticks for nitrates and the presence of white blood cells in the form of leukocyte esteroids have been used. And there have been instruments that can enumerate microbes in broth rapidly. So there are some systems that are out there that can be used. And while I worked at the Cleveland Clinic, for many years we used a rapid screen by a company that subsequently went out of business, and so we lost the ability to use that rapid screen. And when I tried to search through companies, search through the literature to find out what else could we use as a urine screen, I came up with the ones that are listed number 1 and 2, gram stain and dipsticks, but not a whole lot of other methods by which urine screens could be done. And I think that there is reason to at least consider a urine screen. And so how can we do that today? Are there methods that are available? So I'm going to use the opportunity now to talk a little bit about BacteriaScan. New instrument. When we were looking for a replacement for a urine screen in our laboratory, when the one that we were using went out of business, I did run across this company just before I left, actually, the clinical microbiology lab, and got intrigued by it because I thought it had some real potential as a urine screen. Albeit, there's not a lot of labs that are looking at urine screens right now, but hopefully, if you look at the data that I'm going to show you and you think about it in the slides that I've just shown you, you might see the need for a urine screen even in your laboratory. So this is a picture of the instrument, a very tiny instrument. You can batch a whole bunch of these instruments, put them on top of one another, and use them in your laboratory, if you will. I have not personally used this instrument. I left the microbiology lab before it was ready to go, if you will. But although I'm not consulting with the company, I'm not working with the company, I have kept in touch a little bit with the company, and they've been kind enough to be one of the people who asked me to do this particular presentation today. So I wanted to talk a little bit about their instrument. Any of you were at the ASM meeting, you probably saw the launching of this instrument and were able to get a lot more information than I'm probably going to give you today. But the operation principle of the bacteria scan is a forward laser scattering combined with optical density measurements, providing a sensitivity below 10 to the 4th to above 10 to the 10th CFU per ml. And most of you are very much familiar with the fact that when we talk about urines, we're talking about 10 to the 4th to 10 to the 5th or above as being consistent with a urinary tract infection. And so what we want to do with the urine screen is be able to get the results of whether or not in that urine that's been taken out of that patient, I do have 10 to the 4th or greater organisms in that particular urine sample. At the bottom of the slide here, you can basically see the bacterial growth image that's occurring in this particular instrument as it's reading this laser scattering and optical density. And you can see a plot on the right of the concentration changes in the organism versus the incubation time from 0 to 150 minutes. And so you kind of get an idea of where the instrument is going to flag no growth versus an organism like Enterococcus faecalis versus an organism like Citrobacter as is listed on this particular slide. So this is the principle of operation of the bacteria scan instrument and the way it was originally designed for urine screening. There was a study done in Israel, and it's pictured on this slide, looking at a UTI screen in a clinical study design. It was performed at the Rambam Healthcare Campus, a 1,000-bed tertiary Israeli hospital. What the authors endeavored to do in this study is to take a urine and to run it on the bacteria scan 216R, and in conjunction with that, to plate the urine out onto 2 chrome agar media, as you can see on the slide, to also perform dipstick analysis and microscopy. So basically perform 4 different assays on this urine using the bacteria scan 216R as the urine screen that we're analyzing and comparing to the others. 2,400 samples of urine from a diverse population, hospital wards, and collection methods were analyzed. And the objective of the study was to quantitatively compare the performance of various rapid screening methods with a conventional plate culture. So on this slide, you can see the urine was accessed and placed onto the media. A dipstick was used. You're all familiar with the use of a dipstick, whether it be manually or in a machine. The identification of the organisms on the chrome agar were performed in a Vitek. Microscopy is shown, whether that be done by gram stain or some automated instrument. And then at the bottom of the slide, you can see the bacteria scan instrument and the diagrams and the graphs which will come out of that instrument for the results. So what happened with urine screening with the bacteria scan? So on the left, on what's labeled as A, are the growth curves for various gram-positive, gram-negative bacteria and negative samples, as you can see on the bottom with the black. So here's the negative samples down here. If I drag my arrow up to here, here's a coagulase negative staph, another coagulase negative staph, E. coli, Klebsiella pneumonia, Enterococcus, etc. on the left-hand side. So basically what you're seeing are growth curves over that incubation time as compared to a negative, which is less than or equal to 10 to the 4th. On the right, on B, on this particular chart, you can see the additional output from the bacteria scan, showing the optical density on the left and the mean size of the bacteria for 2 coagulase negative staph, labeled as 1 and 2, over 0 to 200 minutes of incubation time. And you can see the growth curves of these organisms and the fact that the machine can then detect that as compared to the negative, we're actually seeing growth of organisms above a particular optical density, above a particular mean size for these two different coagulase-negative staph. So these are the results that one would expect out of the urine screening with using the bacteria scan. So we can definitely see changes in the growth curves, if you will, as these organisms or as these urines are considered positive. So when we look at an evaluation of the pathogens that were detected over periods of time, the statistical performance metrics of the bacteria scan were evaluated at various incubation times. As you can see on the slide here, 30 minutes, 60 minutes, 90 minutes, 120, 180 as are shown. About a 94% sensitivity, 78% specificity was obtained after 90 minutes of resolution. And what we're looking at here is the red bar, or red lines, if you will, 90% incubation. 94% sensitivity, 78% specificity. And you can get a minor improvement, or the authors were able to get a minor improvement in performance observed with longer incubation periods than the 180 minutes that are shown on here. So basically what we're saying is we can actually detect, or the machine can actually detect, the presence of organisms that are growing in this particular time frame. And again, we're talking about only 180 minutes here as compared to waiting overnight, seeing whether or not our urine is positive in growth or not by our conventional methods. So as a urine screen, showing some great potential. On this slide is a summary of the 4 different methods that were used to determine whether or not the urine specimens were positive. And you can see the gold standard, so to speak, agar plate culture revealed a sensitivity of about 86%, specificity of 94%, negative predictive value of 94%, as you'd expect. This is our gold standard method for doing urine cultures. A positive predictive value of 86%, somewhat equivalent to the sensitivity, and an accuracy of 92%. So then if we compare microscopy, which was done on all of these samples, we get a very similar sensitivity, a lessened specificity, a very good negative predictive value, as you would expect with microscopy, but a low positive predictive value. This is a screening tool, and so 46% for positive predictive value, and an accuracy of 79%. Not quite the same as the conventional agar plate culture, but again, it's a screen. You wouldn't expect it to be. What you're looking for here is a high negative predictive value, which it does afford. The nip stick, looking for nitrates and leukocyte esterase, sensitivity of 37% only, so very low sensitivity. Good specificity at 97%. A negative predictive value, a little bit lower than culture and microscopy. A positive predictive value that's actually almost as high as the conventional culture. So when it says that it is positive, the dipsticks seem to agree pretty well. And then an accuracy of 83%. Then if we look at our bacteria scan instrument, which, as you remember, gives us results in a very rapid period of time, not quite so quickly as microscopy or dipstick perhaps, but certainly a lot more rapidly than the conventional culture method. Sensitivity was 95%, very good. Specificity, 80%. A negative predictive value, as you would expect for a screen, that's very high at 98%. So when you can tell a clinician that the likelihood that that urine sample is going to be negative is at a level of 98%, I think everyone would agree that that's really a good result. Positive predictive value is 62%, so you are going to be culturing some specimens, which are indeed going to turn out to be negative when they're cultured, but that's what you want out of a screen. You want it to overcall so that you don't miss any of the positives, and an accuracy of 84%, which is equivalent to the dipstick and a little better than microscopy. So overall, a summary of the results of the study that was done appear to point in a very positive direction in regard to bacteria scan 216. Now, what I want to give you is some data that the company has provided to me in regard to the use of this same, quote, initial urine screen machine as looking at optical density changes for the rapid detection and susceptibility testing in microbes. The data that I'm presenting to you here was presented at the ASM Biodefense and Emerging Disease Resistance Research Conference in January of 2014 by Marshall et al. And so basically what we're talking about here is using this, what was once developed as a urine screen now, to potentially be able to predict susceptibility of microbes that are found in specimens, whether it be a urine specimen or potentially something like a blood culture specimen. And so the use of laser scatter instrument, BacteriaScan, to measure changes in E. coli in concentrations that would simulate a urinary tract infection when these E. coli organisms were exposed to varying concentrations of the drug ciprofloxacin. So the BacteriaScan instrument was used, 37 degrees was the temperature, measurements were made over 5 hours, and the starting concentration of the E. coli was about 3 times 10 to the 4th colony-forming units per ml of broth, which is what you'd expect in a simulated urine culture, if you will. So just think of this as putting E. coli in the presence of varying concentrations of ciprofloxacin. Can the machine predict whether these organisms are inhibited or not, and how does that MIC compare to a conventional MIC? What was shown in the data was there was a successful measurement of the growth rate with repeatability of plus or minus 10% and a 3-minute time resolution. And the instrument was able to approximate a nominal MIC within 90 minutes. And the results were equivalent or compared very well to the CLSI-QC results for that same E. coli organism. So the instrument predicted an MIC of 0.004 to 0.008 micrograms per ml. The CLSI-QC data would suggest the MIC should be between 0.004 and 0.015. Very good results in 90 minutes as compared to a usual 8 hours in an instrument to an overnight manual method of determining a susceptibility of isolated colonies. In addition, some data has been submitted in a poster to ICAC 2015 for the use of BacteriaScan for sepsis detection and susceptibility testing. Donor blood was seeded with E. coli at a level of 1.5 times 10 to the 5th CFU per ml, what would be expected in a bloodstream infection. And 10 mLs was placed into BacTech blood culture bottles, and they were incubated until a machine detected a positive, much as we would do in our laboratories. 4 aliquots were removed from each of the aerobic and anaerobic bottles. 1 tube was not further processed, 1 tube was filtered, 1 tube was centrifuged, 1 tube was lysed, and all of these methods were compared to each other to see what was better, what was worse, did they work, etc. 2 microliters of each sample was diluted 1,000 to 1 and added to Mueller-Hinton broth with ciprofloxacin at varying concentrations, and measurements were made over 5 hours. And the results, growth was detected, and an accurate phenotypic MIC was obtained within 3 hours after the BacTech bottle was deemed positive. Compare that again to 24 to 48 hours when we use our conventional systems. So a real plus, a real possibility. I'm not suggesting this can be used today, but the future of instruments like BacteriaScan and others that are developing in the marketplace are going to provide for us potentially phenotypic MICs that are going to be very comparable to our very long time-consuming phenotypic MICs that we do today, indeed directly from blood culture bottles. I think that's really exciting and something we all need to look forward to in the future. So a couple of slides to end up with. First, clinical microbiology is indeed changing. I give you an article out of MLO here for you to look at your leisure, if you will, to talk a little bit more, to see a little bit more about how are we changing, or maybe I should say how should we be changing. We no longer should be passive. Clinical microbiologists should be now or in the future interventional. We need to communicate, we need to do good testing, and we need to make sure we share the results of that good testing. With our specimens, we know now in the clinical microbiology lab things are changing. We're doing reflex testing. We're doing point-of-care testing. We're doing 24/7 testing in many of our laboratories because we know those results are important to get out as quickly as possible. We're communicating between ourselves and clinicians, ourselves and ID pharmacists, ourselves and anybody else who's involved in antimicrobial stewardship. Many different modes of communication are available. We need to grasp them. We need to do what's best for us, best for our patients. Data management and data systems must change. We can no longer say that we're adapting to systems. We have to have systems adapt to us and to the kinds of results that we do get out of our laboratory. We have a tremendous clinical and public health impact. We need to know that. We need to share that. We need to educate on both sides. Cost versus benefits. Many times, some of the rapid testing or most of the rapid testing we do is going to cost more in our laboratory. But if we can decrease patient length of stay, if we can decrease cost to patient, if we can give them better outcomes, isn't that what we're all about? That is what we should be striving towards. The authors of this article call all of this change in microbiology collaborative medicine, and I think that's a really appropriate term. We need to be collaborators with all of those individuals that we've mentioned throughout this talk. In summary, the use of methods that reduce time to results have proven to be better for the patient and for the medical community in general with regard to patient outcomes, length of stay, cost, and reduced antibiotic resistance development. Rapid methods are available today, and more of them will be continuing to be developed every day. One shoe is not going to fit everybody, but we need to keep abreast of these methods and find the methods that work best for us. Don't sit back and say, I can't do it in my laboratory. Try, educate, educate, educate. Communication is key to using these rapid methods for obtaining the best effective outcomes and achieving the best antibiotic stewardship goals. I want to thank all of you for participating today. I want to particularly thank all of the individuals who have assisted in this presentation. I want to wish all of you successful outcomes. The most important thing in science is not so much to obtain new facts as to discover new ways of thinking about them. I just think that's a great quote for all of us to remember. At this point, I would like to make special thanks to Barbara Lipinskis and APUA for inviting me and doing a wonderful job of planning and organizing. I'd like to thank you, the audience, for listening and not sleeping. I'd like to thank my family for their support. And I'd also like to thank BacteriaScan for sponsoring this in the form of an educational grant. At this point, we're ready for questions.
Thank you. As a reminder, text chat is located on the right-hand side of your screen. To submit a question, type your question in the small text box at the bottom, and when finished, click the send button. I'll now turn the floor over to Barbara Lipinskis to facilitate the Q&A session.
We do have a couple of questions. We're a little low on time, so we'll get to them. We have a request to get copies of the slides. We will make them available. And if you complete the survey, put a little note on there, I'll definitely get them to you. Jerry, for you, what are the confounding variables? Which are the confounding variables removed that change LOS from plus 7 days to plus 1 day or cost $70K to $8K?
Yeah, for sure. The article is very complicated, so I urge you to take a look at it if you'd like to. It was the Nardell article, but basically they were looking at taking and removing the confounding variables that would be more procedures done on patients. Were there more costs before the onset of the infection so that those could be removed out of the cost? Were patients on catheters? Were there comorbidities? Were there other underlying conditions? And there are some others, and they do a really nice job of explaining how they matched controls and then how they took out some of these confounding factors. But basically, the procedures, the higher costs that may have been ensued before the infection was even determined, and the use of catheters were 3 of those confounding factors.
What is the cost per test for the bacteria scan test? Do you know?
I actually do not know that. I asked Rhonda before, one of the representatives from BacteriaScan, if she'd be willing to do that. I'm not sure they're ready with a cost per test as yet, so I can't answer that question. My apologies.
Okay. And where would you recommend a small institution start with their administration to open discussion about these options? Good question. How do we get started?
That is such a good question. So I think all of us in the clinical microbiology lab have to be a part of this, but let's face it, a smaller contingency or the director of the lab is going to be the one that's going to be going to the administrator and saying, we really need to use Verigene, we really need to use fish probes, et cetera, et cetera. So I think what you need to do is get a battery of outcome studies together that you could share with your administrators. I hate to say that cost is going to be a big part of it, but it is, and we all know that. We want to show that we're going to have better lengths of stay, that even though it's going to cost more for us in the laboratory, look what it's going to do for our patients, if you will. So you want this small cadre, if you will, of outcome study to go with you. You want to show them how much time it takes to do the method you're using in your laboratory now, and then you want to compare that. One of the perhaps great methods that you can use for starters with your administrators would be multi-talk, because I think it is something that a lot of institutions are looking at, and that might be the best way that you can start getting your administrators geared up to know that you're going to be coming to them often and asking for the use of some of these instruments, and you're going to be coming with some good data that shows they are really going to be effective in your lab.
Just have time for one more question here regarding the bacteria scan work. Are there yeast studies underway for those tests?
I actually don't know the answer to that. I feel really stupid in saying that to you, but I don't know the answer to that. Perhaps Melinda could ask, or individuals could contact Bacteria Scan at their website and get that information, if you will. But since we know that yeast are indeed in urines, I am quite sure that Bacteria Scan does have data with regard to yeast.
We are just about out of time, just slightly over our 90 minutes. Several people have asked if they're able to get copies of the slides or copies of the presentation. The answer is yes. Everybody who registered will get an email, and there'll be a link there where you can download this. So we will be making this available for everybody. And I think that's pretty much tying it up for us now. Okay.
On behalf of APUA, I'd like to thank you for your participation in today's event. A post-event survey will appear requesting your feedback. This survey will also be sent in a follow-up email to be completed at your convenience. Please take a moment to complete this survey, as it will help APUA plan future web events. This does conclude today's program. Thank you and have a great day.